made key contributions to medical image analysis and modeling, and I'm very excited to hear what the future of healthcare has for us.
Thank you. Oops. So thank you, Nick. So Nick asked me to say a few words about the Christabel Pankhurst Institute. So I'm the director of the Christabel Pankhurst Institute, which is the Manchester Institute on Health Technology Research and Innovation. Is a joint initiative between the University of Manchester, all three faculties, also Greater Manchester Combined Authority, Manchester Foundation Trust, Health Innovation Manchester, and Bruntwood SciTech, which is kind of a state's organization. And the main goal that we have in the Institute is to contribute in Greater Manchester to create an environment for innovation in health technology. And in particular, the way we interpret health technology is a focus on AI and digital health, is one of our programs. The second one is in computational medicine and biology that I'm going to talk a bit today. And the third one is in advanced diagnostics and therapeutics that also touches a bit upon what we do. And our goal is really to focus not only in the developing of the technologies, but in their translation to our NHS partners. And Christabel Pankhurst was an alumni from the University of Manchester, and many of you would recognize the name Emmeline Pankhurst from the suffragette movement. So Christabel was one of her daughters and the only alumni from the family from the university. But they, well, as you well know, they embodied the changes of the time and bringing women's vote, but also more broadly in Manchester, there is this passion for social transformation as well that characterizes our region. So what I'm going to do today is I'm going to talk about the concept of in-silico trials in particular as one aspect of the future of healthcare. So the thing that we are trying to do in medicine, or the things that clinicians have to sort of commit to when they get their title, is what is called the Hippocratic Oath. That is that the first thing clinicians need to do is not to do harm, okay? And that's the first rule, so not mess it up. In particular, when we think about medical products, so whether they are pharmaceutical products or medical devices, there is a very lengthy process to go from discovery all the way towards generating the evidence to go to market. And this is based on solid pillars of scientific evidence. I mean, regulatory evidence, I'm going to talk a bit about regulations, and regulatory evidence is essentially scientific evidence. And it's scientific evidence about the safety and the efficacy of the medical products. And the way that we normally come to assess those is through bench testing, through animal testing, and through human testing. And what is very important to recognize is that all these three ways are models. I was given a talk once and someone said, no, randomized controlled trials, that's not a model, that's real data. I said, well, no, it is a model, because the model that you're making is that your assessment in 100 people or 1,000 people is a representation about what's going to happen when you go to the whole population, right? And that we know is not necessarily the case. Now, is it really so solid? And this is partly the motivation of this talk. So first and foremost, it takes forever. So if you are in a medical product, a medical device, or a pharmaceutical, can easily take a decade to get all the way from discovery to the market. All of these different sources of evidence that we talked before, all of them have limitations. If you do testing on bench, you don't replicate necessarily physiological conditions. If you are measuring on animals, that not necessarily translates to humans. Doing experiments or trials on humans has all kind of practical and ethical limitations that induce, among other things, biases on the evidence that we produce. And of course, you can do most of the things wrongly as well. Just to show that it's not working, this is what is called the implant files study, which is a journalistic study that happened from 2008 to 2018, and it took about 10 medical products and looked at the evidence of harm that they produce. 83,000 deaths, 1.7 million serious adverse effects in the US alone. And if we say, well, but that's the US, well, we have the Cumberledge report in the UK. There was an independent review on medicines and medical products that effectively studied, several decades of harm that was inflicted on people due to devices and products that are the same that in the other study. We're all going through the regulatory process that is in place at the moment, which is the one I described before. So these are all FDA-cleared products. Just to show an example, this is only a few weeks old now. This is a left ventricular assist device. This was taken out of the market. It's an Abbott device, and it produced 14 deaths, 273 serious adverse effects. And you say, well, this is this company that has a problem. Actually, if you look at just 2024 so far, this is, I think, I checked last week, and it's about 70 of them. This is a few weeks old now. But there were 50 recalls halfway through the year. And this pie chart, what each color, this wedge, each of these wedges indicates is a different company that had a recall this year. The bigger wedge is 4 recalls, and then the thinner ones is 1 recall. So it's a problem across the whole sector. The other thing that is problematic is the sustainability of this system. So if you look at what it takes to go from discovery all the way to the regulatory evidence and post-market analysis, there is a 90% attrition rate, which pretty much replicates what happens in the pharmaceutical space as well. The average cost of producing a complex medical device is about £54 million. And this is the average cost. You can see there the bracket, which is quite broad. And this is if you get it right first time. But the actual cost, if you incorporate all the capitalization costs, so all the mistakes you do in the way, is about 10 times that. Okay? So one of the things that I'm trying to think about in my Royal Academy chair and in an ERC Advanced grant that I started in December, is how we can change the way that we do the testing so that we can reduce these 500 million as close as possible to the 54, and wouldn't it be great to halve these 54 to half of them? Now, the thing that really hurts me, I'm an engineer by training, is this slide. This slide shows the 5 top FDA identified root causes of failure of medical devices, and the number 1 is device design. So if this was the aerospace, the automotive sector, the manufacturing sector, you will fire the whole department of design from these companies, right? 53% of all the class 1, so the most dangerous devices, the implants, 53% of all the implants have fundamentally a problem on device design. And you see all the other reasons are secondary compared to that one. So for me, first do not harm equates to simulation first. We shouldn't be putting our devices even in a bench, because that produces waste, or in an animal, because that compromises animal welfare, or in humans, let alone, because that puts individuals at risk before we have actually done our homework testing on models and on simulations. This is exactly what we are doing in other sectors. When I show this sort of presentation to people from the wider engineering, they look at me and say, what's new? Because we've been doing this in the aerospace for 20, 30 years, right? So, and you could say, well, but aerospace systems are not so complicated as healthcare. I challenge you on that. They're multi-scale, highly regulated, mission critical. They are full of uncertainties, right? So they have a lot of the similar characteristics of the human systems, the biological systems. I'm not saying they're identical. I'm not saying that is easier, biological systems. I'm just saying that we shouldn't, dismiss outright these ones. And they're going to a transformation, the digital transformation, to actually reduce the amount of physical tests and going more and more into virtual systems. If I show you 30 years worth of innovation in the automotive sector, you can see the difference between a product 30 years ago and now, right? And it's not just the product that is different, the processes that we follow are different. So we don't do wind tunnels anymore. We do aerodynamic simulations. We don't do physical testing and physical crashes so often. We do virtual crashes. And we do, we utilize AI more and more. If I show you 30 years worth of development in a product that we're going to be talking about, it's still a bunch of wires, okay? So this is a flow diversion device for brain aneurysms. So we're going to talk a bit more about this, but these are vessels in the brain that bulge and can blow. And these are coils. These are stent-assisted coiling, and this is flow diversion devices. But the material pretty much hasn't modified over 30 years. And you can still have nearly 20% complication rate, 3% death rate. And we can do exactly what we do in the aerospace. We could actually see through models what is the prediction regarding, for instance, hemodynamics, and we'll talk about more later clotting as well. So computational medicine, is essentially the use of engineering, mathematics, and computer science principles to understand better disease and design better therapies. And what in silico or in computers tests, if we're talking about preclinical, or trials, if you're trying to replicate clinical trials, they are sophisticated computer models. Some of them are AI models, some of them are more mechanistic, and we'll come back to that, that actually use detailed prediction models that can, in very controlled environments, and in conditions to actually test and understand the operational regimes of these devices. And there is quite a number of recent documents that we have produced as part of the sort of work that I'm doing in the academy, like this one, and this is from the Reagan-Udall Foundation for FDA that supports FDA development. The other thing that is unbelievable is that when I took this morning a flight, so I came from Hungary from another meeting. When I took my plane, I knew that I had 1 in 10 million chances of having an accident this morning, right? If you now participate in a clinical trial, you have absolutely no idea what is your risk of dying by just participation, assuming you're not in the placebo arm, okay? And actually, all the evidence in the literature is that you cannot even calculate that probability. Okay, we don't record the data in a way that you can calculate that. So that's crazy. So what we are trying to do with in-silico methods is not to eliminate completely the need for doing all the things that we normally do, but it's another tool in our arsenal, in our armory, for actually produce regulatory evidence. So if you think about this diagram, the horizontal axis is the life cycle of a medical product from discovery to post-market monitoring. And the vertical axis is the number of problems in your design that you identify and you need to fix. So what we do at the moment is this dotted line in dark blue, which is we do very little testing early on. Why? Because we don't have any money. So if we are a startup, we are desperate to get some capital. And if we get 2-3 million to get started, that probably will be spent in our animal experiments, which are about 100,000 pounds per animal, if you're thinking about a cardiovascular device. So you do 20 animals, you spend all your money, right? So you need to freeze your design space very quickly so that you get some evidence to go back to your investors and ask for more money, right? So by the time you get this money to then go and do more preclinical testing and regulatory approval, you are deep down into this line. And the deeper you go, the more expensive it is to go back and do any changes in your design, right? And if you are lucky, between quotes, and you're allowed to go all the way to do a human trial and then something goes wrong, you're in deep trouble, okay? So what we are suggesting is that if we were able somehow to replicate conditions similar to the ones we will have in the end, early on in our design process through some sort of digital means, I throw the word digital twins here, but it's just one aspect of it, so that we could actually figure out everything that can go wrong much earlier on and fix it. By the time we get to do the trials, we have the risk, the product, which means better, good thing for the investors. We have the risk, the patients, because the likelihood that we will administer a therapy that is problematic has been reduced, and also we have saved a lot of money on the way. Okay, so that's kind of what we are trying to do. Something I said earlier is that in a bench, animal, and human, and I would add now in silico, are models. All 4 of them are models. And it's not a question whether in silico will replace everything else. It's why on earth would you justify yourself to use only 3 means of investigation if you have 4? Can you actually use this 4th avenue and then effectively build a regulatory dossier and build sort of a regulatory evidence that is taking into account all of those elements? And I don't have time to go today through the developing standards in this area, but they're all risk-based standards that actually try to weight the influence of the models in the decision that you're going to take, and also whether you rely fully on the model or you rely on various sources, and also what is the impact of that decision that you're going to be making on the base of that device. Is this something that makes a difference? Yes. So this is a case study from Medtronic, I think to remember. They went 2 years earlier to the market by being able to use these sort of techniques. They reduced by over 250 subjects their trials. Just to give you an idea, the cost of a single patient in a randomized controlled trial, the average cost is $40,000. So multiply by 250, that's what they save. And these costs can be between 16,000 to 120,000, depending on the type of device you're talking about. They saved 10 million because of the reduction in trials, but also they went 2 years earlier. So that means you have a lot more devices you have implanted and sold during that period. Okay, so what does this paradigm shift involve? Well, it requires to build virtual patients and virtual populations. It also requires to have models of the interventions and the examinations you're going to do, and you also need to have virtual readouts and virtual outcomes. And every single one of these steps has to do with the use of computational modeling and simulation, and a large component of that is also AI and prediction models. So how you build these models, and what is an in-silico trial? So an in-silico trial is a massive simulation space, like an ensemble space like this, where you have certain bundles of dimensions that represents anatomical, mechanical, rheological envelopes, and other ones that represents lifestyle, physiological, and operational regimes of your product, and then another one that represents all the design space on the parameters of your model as well. And all of those things can be obtained nowadays with the availability of digital data at scale from regional data sets. I'll talk a bit about that in a minute. So a dot here is a particular anatomy in a particular set of physiological conditions for one particular device, and this is a virtual experiment. This is the basis of observations in silico trials. Okay, so let me give you some examples of these and the focus, and then I have a second part where I'm going to show you some of the things under the hood to give you an idea about kind of where the AI roughly is. So I work fundamentally in two, you need to focus in your work in some areas to be able to show these concepts all the way through. And I've been focusing for about 20 years in flow diversion devices for aneurysms and coiling. And the ERC that I got is on TAVI devices, which is a prosthetic valve for the heart. And these are the two main areas where I'm working at the minute. But I'm going to present today all the work more related to aneurysms. And maybe in the questions I can explain a bit more if it fits some of the work that we're doing on the heart. So the first, I will start by the end, in a sense, a study that puts it all together and that also tries to make a number of points. What is what I call an in silico reenactment of a real trial. Second is how you can combine physics, biology, and physiology into one of these trials. And also that you can have emerging behavior, so things that you don't model explicitly, but that come as an emerging property, basically, from your simulations. And that shows that actually you can do more than just purely replicating the knowledge you have. So first a bit about aneurysm. So an aneurysm is a bulge of the brain, of the vascular vessel of the brain. If this bursts, it produces an hemorrhagic stroke. And this is lethal in 1/3 of the times. Another 1/3 of the time you have high morbidity, and then a 1/3 of the time you might be able to survive it if you catch it early on. These aneurysms love to appear in bifurcations, and this circle that you see there is what is called a circle of Willis in our brain, which is a smart system that we have in our brain, so that if one of the vessels becomes closed, you get contralateral circulate flow, and sort of, your brain continues to be fed. There are a number of techniques to treat them. The oldest one is what is called clipping. So it requires craniotomy. So you need to open the skull, put a clip, and then put it back. And as you can imagine, this is a quite invasive approach. And then all the other ones are what I call the plumber way. So you go through the groin, through an incision in the groin, under catheter guidance all the way to the brain, and you insert coils with the stents, especially if they're wide neck. And then people figure out that you could use, rather than stents, flow diversion devices, which are more tighter struts, and you don't need to put a coil anymore. And fundamentally, what all of these three guys do is they produce an stagnation of the blood flow that promotes clotting within the brain. And that is what selectively closes the aneurysm. But there is a whole zoo of these devices. And each of these guys requires a trial, animal experiments, and I took out another slide that I have, but there's about 40 of them, different products in different stages. And Also, if you actually look at these products, there may be 5, 6, 7, 8 years in the market, and then something else comes and overtakes it. So if it takes you 7 years to design it, and you're only going to recover your cost in 7 years, that tells you why they're so expensive. A device like this might be easily cost between 5,000 and 15,000 pounds, right? It's not the material costs, it's all the R&D that went into that. Okay, so how these devices perform. And this is a device from Medtronic. It's a flow diversion device. So basically, you have the blood flow. It goes into the aneurysm. It keeps pushing and pulsating and potentially bursting it. So you put these coils that reduces the blood flow. It produces clotting. And basically, you remove the aneurysm from the circulation. But you have small vessels called perforators that are the ones that feed the brain. So you don't want to close them, because otherwise you have an ischemic stroke. Okay? So you need these ones. To remain patent, and that's why this is not like in the aortic aneurysms that are grafted stents. So these are just bare metal stents. Okay. So the first question that we try to answer, well, a question that we try to answer, wasn't the first, is can an in-silico trial replicate and perhaps even expand what we know from conventional trials? And we set off to study that device that I explained you before, because the company claims that this is the most studied flow diverter in the world. What does it mean? It means that they did a pivotal trial. The pivotal trials are the trials upon which you go to market, which is published in Radiology. It's this one here. Whooping number, 106 patients, probably 30 to 40 million pounds, 8 years from design to publication. You need to recruit patients, follow them up for 5 years to see the outcomes. And that's it. That's your model. 108 patients, and we model the rest of the world with them. Then this is for large aneurysms, and then they say, what happens if we put these to small aneurysms? So go and you do another trial called Premier for small aneurysms. And then they went to the market, but then they had to keep a registry, which is called the Aspire registry, which is patients that you're already selling, but you keep a close watch on what's going on with them, and you record every single potential failure with those. So if you put all these 3 studies that we're going to compare against together, you don't have 500 patients. Okay? Okay, so that's the device, how it looks. And a trial basically is the following. You take a library of anatomical models. You then deploy the device virtually using computational mechanics in every single one of those anatomical models, and then you need to come up with some sort of surrogate endpoint of outcome, of performance. Remember that I said that there are 3 ingredients: the virtual populations here, the virtual intervention in this case, the deployment of the device, and the readouts, which in this case is something that we call the reduction in the maximum time average velocity at the neck. So basically, we look at how much of the flow that went into the aneurysm is removed out of the circulation after you put the stent. And you see that the same device, the same product, 2 different patients. In one, we reduce 64% the flow, and therefore we're going to clot it most likely. The other one is only 27%. Now we do measurements with wearables, and we say, okay, what happens when I look at a number of patients, in this case 100 patients, and we look at what is the average waveform in the carotid artery and the variability across multiple subjects. And because we did a Holter examination, so in 48 hours, we know what is the average resting conditions and the average exercise conditions from these patients. So the rest condition is the blue, the stress conditions is the red. And you can effectively take every single one of these guys, and it's the same anatomy. You put the same device, and you just look at what happens between rest and exercise, and again, different performance. And then finally, you don't need to do randomization here. You can put every single device in every single patient, so you can do head-to-head comparisons between the performance of 2 competing devices and look at the outcomes of each of them. The Pipeline, that is the one we studied, and the Enterprise, which is another manufacturer. Now, when you do randomized controlled trials, manufacturer 1 does in a population, manufacturer B does in another one, and then you need to compare the conclusions of these 2 studies. I wish you luck, okay? Okay, so what we did in this publication is precisely what I just explained, and we show what it says on the tin, that in silico trials of intracranial flow diverters replicates and expands in size from 3 conventional trials, the one I showed you before. So let me explain you roughly what we did so that you understand a bit the methodology behind. So first, we need to define a population. And in this case, we did the simplest thing you can think of, which is an enumeration of subjects. So we started with a database of 1,000 subjects, and we imposed all the inclusion and exclusion criteria of those trials, and we ended up with 88 subjects when you do that, because that's what they had. And then we compare FDPAS, which is our in-silico trial, with the 3 other trials that I mentioned before. And, well, I can tell you that working with an epidemiologist and a trialist, we effectively satisfied ourselves that those groups are exchangeable, that we could use that one. And we didn't have access to the trial data, so we had to create a population that had similar characteristics. Now, the other thing you can do then is you create from a different set of subjects, because we didn't have the waveforms from the subjects that we had the images, we had the physiological boundary conditions, and you effectively do this nice animation. What it tries to say is that we try to learn the joint distribution from the 2 marginals, effectively. So you do kind of the multiplication. And are they independent? No, they're not most likely, but because we are interested in, our problem is that normally we under-test. What we are actually doing is over-testing with this, so we are less concerned about the independence here. So every subject is one anatomy with one set of physiological conditions, and we then have to do a virtual implantation of the device into each of those. And again, there are techniques to do this effectively and fast, but you can do that with computational structural mechanics, for instance. Then you need to solve the physics, in this case, the Navier-Stokes equations for blood flow, and you actually show what happens. So this is particle simulations. So basically, we put particles there and we let them flow with the blood flow that we calculate using solving Navier-Stokes. And what you observe here in this aneurysm is that the particles stay longer than in here because you increase the residence time because of the slowdown of the stent. Then you also can put the different regimes of rest and stress, and you create conditions of what we call normotensive patients and hypertensive patients, representing the pre-flow diversion implantation and the post-flow diversion implantation. So we have pre and post rest and stress conditions. Okay? And then finally, you can solve the biochemistry as well, and you can solve the clotting process. So what you observe here is a metric that tells you where the clot is being formed and the quality of the clot as well. I'll come back to that a bit later. But what you see here as well is that in the normotensive case, the clot stays in the neck, while in the hypertensive clot case, the clot gets into the side branch, and that's trouble. That means that you will have an ischemic stroke there. And I'll comment that in a minute. And then once you're done with that, basically these images that we have simulated are like virtual images, and you can do metrics and do statistics and do things like in any trial. Yeah? So if you do the comparison, for a moment, take these two, and you do the average of these two, and if you do, it gives you around 75. And I'll tell you what is each of these in a minute. But if you take the average, 75 is spot on the study from the other 3 trials that we had before. What this means is that we assume that the 88 subjects that we had were all normotensive, and we also assume that they were all hypertensive. And if you just change the physiology of these patients, you have 15 points of difference in outcomes, which effectively means that this device perhaps is not just the device what has an impact, it's also the conditions in which the implantation is being made. And we'll come back to that in a second. We also looked at failure scenarios that are only anecdotally reported in the literature, like what happens when you have a side branch, what happens when you have large and giant aneurysms, and what happens when you have high aspect ratio aneurysms. The aspect ratio is the length of the aneurysm divided the neck of the aneurysm. So we could demonstrate for each of those cases what are our predictions using these models, this performance. And I also mentioned before, there are two interesting things here, but I'm going to mention only one. So in this case, the case 3 and the case 4, you see that it's exactly the same anatomy, the same device, everything is the same. The only thing that is different is that this is a normotensive phenotype, this is a hypertensive phenotype. This is the one that generated the side branch clot, while the other ones didn't. Well, from the 88, we had 27 subjects that had the side branch, and in every single one of them, we observed this effect. Okay, and this is something that is known in the literature. Well, sorry, there is anecdotal evidence in the literature that if you have a side branch, you don't put a flow diversion device. We believe that the problem is not the device, it's the combination of the device and the interplay with the physiology of that patient. So it may be that if you actually treat all of these patients with a hypertensive prior to the treatment, they may potentially perform better. And this is what is called a combination therapy, and it's super complicated to do trials because you have two factors, the device implantation and the pharma, the drug that you're giving to them. And it's a combinatoric, study. Okay, second example, and these following examples are going to be a bit shorter because that one I introduced most of the concepts. This is what happens when you have incremental innovation. Say you already develop a device, but you want to see whether that device works in what is called off-label conditions. So the label of a device is where it says this device is to be used in this, and this condition. When you use it off-label is when you're not listening to that and you're nonetheless going forward and just applying it. For instance, because you don't have any other device for that type of patients and you need to do something with the person that you have on the table, right? So this device, the device that we were talking about, has only been approved by FDA in this portion of the vasculature. All the trials are just for this, and it turns out that the aneurysms that are in the posterior communicating artery are 25% of all the aneurysms, right? So that's 25% of the market that you didn't cater for. It turns out that those, there are different variants, and we'll mention that in a minute. There are variants, so this is the circle of Willis that I mentioned before. This is what is called the non-fetal variant, which is the standard one, but you have two types of partially and fully fetal variants, which is where this branch is hypoplastic, so it's small, or it simply is absent. And these are not ill patients. These are just patients these are subjects that have a different type of anatomy. Well, it turns out that when you have a missing artery or a reduced artery, there is a change in the epicoma flow and also in the diameter. That, of course, changes your characteristics. Now, these two cases, or these two situations of partially fetal and fetal variants are more prevalent in black people than in Hispanic and white people. So if you look at the ratio between these two and the peak, is much larger than in these other ones, or more smaller in a sense. Well, it turns out that if you do the trial, and I'll save you the whole exercise, we did a study to look at what happens in the performance of that variant compared to the standard one. And also, this was our primary hypothesis, that there was a maintained patency, and also understand the influence on the flow in this artery with aneurysm morphology and treatment efficacy. So if you do the whole study, and I'll save you all of this, what you observe is that the fetal variant, so the one that, certain ethnicity have more overly represented, response half the efficacy of a conventional, of the white population. So this is an example where we have evidence of health inequities induced by the type of treatment that we have. So we know that now we can do something about it. And as you can imagine, certain demographics are more difficult to involve in certain trials. And many of these trials, will then be used for treating people, in other geographies where their representation is more prevalent. Okay? We also, by the way, show that in a number of metrics of anatomy, there is no change in the reduction in flow. So you see that it's more or less a flat line for any of these variables. So effectively, this device is not so much affected by any of these geometric properties that are thought to be important. The third example is how you can model things that are difficult to model and that are related to the operator influence. And I'm going to change of device to coils instead of stents. So coils are what it says on the tin, sort of shape memory materials that when they are deployed within the aneurysm, they sort of reconfigure in 3D to fill in the whole volume. And what you see here is before you start filling in and after you fill in, and also here there are some specimens where you have a cross-section and you see all the struts of your coil. Some of these coils have hydrogels and materials that promote coagulation as well, and you can see also how the shape memory helps to do this filling appropriately. And again, there's a zoo of them, another number of devices. And we know that the outcomes are depending on the skills, on the type of coil. So one important metric here is what is called the packing rate, which is the amount of metal in relationship to the volume of the aneurysm. Okay? So this is the way you start, and by the time you finish, you may have 30%. It's difficult to pack more than 30, 35%. So when you do that, and that's a surgical specimen, you can see all the struts, and then you can see also in histology all the struts coming in. So we developed algorithms based on solving Dijkstra and minimum cost path with some constraints, where we actually can incorporate some heuristics about how this coil goes and fills the volume, and then how you validate those models. I'm saving you a lot of validation behind everything we've done, but just to show a bit the sort of things we've done. So one example is with rabbit models, where we can use animal experiments to validate our computational models rather than to try to design the device itself. So you use the animal models as calibration, and you reduce the amount of them that are required. So for instance, in this case, we implanted these coils in actual rabbit models, and then we did a virtual histology of our coiling system, and we look at the distribution of the coils in the animal versus the distribution of the coils in our computational model. And we show that they actually distribute in a very similar way than in the actual model. And the other thing you can do is you can have an actual physical model that you, like a acrylic model, that you put into a scanner, and then you can flush contrast agents and solve the time dilution curves of those models and look at the wash in and wash out in your scanner. And you can do a virtual angiogram as well by solving the transport equations of the contrast in addition to your blood flow simulation. So what I'm showing you here is the actual measurements with in the phantom against the simulations of the time dilution curves on our computational model. And you can see that they're pretty much spot on. Okay, now your question probably is, what are you going to simulate here? Because these devices, as we push them and pull them, they may end up anywhere, right? Well, the beauty is that when you don't know something, what you can do is sensitivity analysis, and you can look at how important it is that you would know those things you don't know about. So what you observe here is, this is like an stochastic fill, right? Like a random fill. So the vertical lines corresponds to 20, 25, and 32% packing rate. And each of the horizontal lines is like a realization of that random fill. So I call it configuration A, B, and C, but I could give you 50 if you want, right? This is equivalent to put the coils to 20%, then I take them, I put them again to 20%, I take them, I put them again, and the only thing I keep constant is the packing rate. Of course, they will end up in slightly different positions, mimicking what would be different interventionalists. And you can do that for different percentages. And by the way, the coils, every color represents a coil, and because we know the lengths, we know the diameter, we can calculate the volumes. So if you do that, and then you show what happens as you increase the packing rate, what you can do is functions of the amount of flow reduction as a function of the packing density. And what you observe is that there is a point beyond which it doesn't matter that you keep putting coils, because you pretty much obtain the asymptotic behavior of that flow reduction. If you keep putting coils, A, you're wasting money at the rate of 1,000 pounds per coil, probably. And second, you're most likely putting the patient at risk because you're overpressurizing that aneurysm from within. If you put less than that optimal number, then you have another problem, that is that different people will end up having different outcomes because you're in the regime where actually it matters the way you put them. So there is that sweet spot. Now, interesting, about 10 years before that piece of work, there was another paper based on real patients where they show that stable aneurysms have packing rates or embolized volumes which were higher than their corresponding unstable aneurysms version. Okay, and that they were for small aneurysms or for large aneurysms, and depending on the neck, different optimal numbers, of course, because they're pergene specific, right? So you could develop a chart that, given the location of the aneurysms, the shape of the aneurysm, and a number of other characteristics of the patients, gives you the optimal packing rate. And in a sense, that can become part of the policy in which you treat in your hospital. Okay, very quickly over some of the methods so that you know roughly what is the technology sort of things. And I would say, as a bit of a philosophical couple of slides, and also to try to explain how this all connects to AI, there is, well, maybe some of you are familiar with this DIKY pyramid. But we collect lots of data, but ultimately what we want is wisdom. Wisdom is validated knowledge upon which we can take decisions about future scenarios, right? So having lots of measurements is useless if I still don't know what to do with the patient. So the wisdom is that validated knowledge. Knowledge is a structured information, and information is processed data. Now, there are different ways in which you can have you can do models. You can go data-driven, so bottom-up, or you can do mechanisms-driven or principles-driven, which is top-down. And you can also do middle-out, which is what we tend to do, which is when you basically have some things that you know a lot and some things you have no clue. In some cases, you have lots of data. In some cases, you have a lot of prior knowledge. So this is basically the whole idea of kind of my philosophy behind the work is to use suitably phenomenological or data-driven versus mechanistic or principles-driven models by, processing information and modeling the knowledge there. Why this is important? Because phenomenological models are limited by the data you have and are limited by your sampling rate. And I think the previous talk showed that depending on the way you sample, you get different views of the world. Well, this is the same. Depending the way you sample your data, you will have different models potentially. While mechanistic models are limited by the knowledge, the basic knowledge that you have and your theories, effectively. There is an interesting paper by a guy called Dirk Helbing in ETH that he argues that in the next few years, while the processing power will continue to grow, like the Moore's law, and the data volume will continue, exponentially, doubling every 18 months, the Moore's law. Data, they say that is doubling every 12 months and that there will be a not-so-distant future where it will double every 12 hours, right? But it's still exponential, and two exponentials have one crossing point, right? And then the other thing is that the systems we are trying to model are so complex and there are so many interactions that the best way, he argues, to model it is with a factorial process. And a factorial function overtakes any type of exponential function you can think of. So the point is that there will be a moment where the complexity of the systems we are trying to model is much bigger than any data we will be able to amass and any computational power we will ever have. And the only way to break this curse is by actually incorporating prior information, right? And this is the whole idea about, combining, in some cases, you have a lot of knowledge and you should use it and not use data when you have the knowledge, but there are things that you simply don't have the principles yet, and there you need to use the data appropriately. Okay, so that's a long way to say that sometimes we use AI, sometimes we use principled models, and we don't, and I don't have a problem with that. I think it would be wrong to use deep learning techniques where you have the principles. So this is an example of a pipeline. You go from data at scale to images, and you create three-dimensional models. You tend to use a lot of deep learning here. Then you need to generate numerical meshes. You need to have models of the structural mechanics in this case. Gaussian process models to model, for instance, things like the physiological variables. Then, again, mechanistic models of the physics of the blood flow, and mechanistic models of the reactions for modeling the biology of the clotting process. Practical challenges, as many as you want. So sometimes imaging is not isotropic and nice. Sometimes you need to deal with the fact that data has overlays or is close to the resolution limit, what you're trying to image, and you have missing vessels or kissing vessels. The other thing is you want to do this at scale with several hundreds and thousands of these subjects ideally. So, techniques that we used to use 10 years, 20 years ago, they simply don't work anymore. And this is where deep learning techniques tend to be actually quite good in general. But the other thing is you don't just do segmentation. You need to compute center lines. You need to identify automatically what are the inlets and the outlets to set up boundary conditions. You need to be able to know where the aneurysm is, where to deploy the device, how to do meshing. And then ideally you want to do quality control in every single step here. So there's a whole praxis behind how you do this properly. And we do a lot of validation on the way, which I'm going to save you for today. The other thing is I talk about some of the physiological variables. So we have also coupling. So you have these detailed three-dimensional models, but you can't model the whole body this way because it takes forever. So what we have is models based on wearables or physiological variables, and then different levels of dimensionality reduction, like using systemic models as electric equivalents, which are 0D or 1D, and those can be coupled to the areas that we are interested in, the 3D models. Then we also talk about, say you do the measurements of the physiological variables, and we also develop models, in this case, where Gaussian processes models, where we can replicate, for instance, the performance of what is the scaling factors for these waveforms, depending on your body mass index, your age, your sex. And you can then generate regimes, like what happens, for instance, for multiple subjects. But you do that in a way that is matched to the properties of your patient. It's not just a random number that you're giving to every subject. And also you can create different levels of inter-subject variability that may replicate different conditions of stress, for instance, or stress levels in this case, or different subjects in this case. The other thing that we're starting to work is in using deep learning for accelerating some of these sophisticated physics. And again, I don't have lots of time to go over that, but there are things called, there are ways to accelerate physical simulations that incorporate some of the challenges that we observe in here, the fact that they are nonlinear processes, they are complex geometries, they're multi-physics, they're multi-scale in time. So you want to look at what happens in several days, but at the same time you have a loop every second for every cardiac beat. So how you model all of that. And then there are techniques like reduced order modeling techniques that allows you to find basis functions where you solve the flow in a space and time. And we've done work where we looked at, for instance, either vessel segments or aneurysm segments, and using sort of neural networks find ways to essentially replicate. So this simulation here is probably 2 orders of magnitude faster than this one here. This is a full order simulation. The other one is an accelerated network that also has incorporated some physics constraints so that certain physics properties are maintained. We talk a bit about the clotting process. So there are some biochemical equations that we need to conserve. And we also do validation again experiments where, for instance, we observe, so this is a bit unhelpfully colored in the sense that the redder it is, the clot is called white clot, and the whiter it is, the clot is called red. So a red clot is a clot that is jelly and redilutable, while a white clot, it has a lot of fibering and other materials inside, so it sticks, right? And you want white clot, not red clot. So you see the clot here is strong, and this corresponds to this bit, and in here corresponds to this other bit here. So it replicates what we've seen in experiments. And then the other thing is, if you only use data from trials to actually test devices, you're going to be biased because probably many of you know that when you do a randomized control trial, a typical problem is that you have a selection bias, right? So if you want to look at what happens with patients outside of your trial space, you need to find some way to generate patients in a non-trial conditions. So a beautiful opportunity for that is UK Biobank, right? That has a very large cohort, which is less constrained than a randomized control trial. Now, the problem with UK Biobank, or not so much of a problem, is that because this is a volunteering trial, they're not going to be doing anything that is invasive, like putting contrast to your brain, to your blood. So as a consequence, we don't have angiographic scans from UK Biobank. So what we develop is a technique that you take non-angiographic imaging and you can generate angiographic scans. And just to do a bit of a Turing test with you, so one of these two is a real scan of angiographic data. So this is not UK Biobank, but one where we had the two modalities. The other one is synthetically generated from only T1, T2, and proton density imaging from the same subject. Okay? And this is a similar view, but this is a slab view. So the one, can anybody guess which one is the real one? Sorry? No, one of them is real and one of them is synthetic. So this is the synthetic one, okay? I don't think the lighting is playing in your favor, but I can tell you it isn't easy to figure out even if you have the lighting in your place. So we've done a lot of work by comparing a number of metrics, and perhaps this is the most useful one. So this is the ground truth, and this is the synthetic, and we have done the segmentations and then look at the fluid dynamics, and they're pretty much indistinguishable in many ways. Okay? So we are doing a bit more detailed studies because I'm sure that there are ways to break it down. But it shows that you can, imagine now we have the full UK Biobank and we can synthesize all of their vascular trees. Then you have now all of a sudden trials with 80,000 subjects that you could run. Okay? Okay. In conclusion, so hopefully I convey the idea that there are problems with the current regulatory processes and innovation processes, that modeling and simulations is, I think, an essential new ingredient in this space. But that it's not only a technical, I haven't covered that actually in a lot of detail, but it's not just a technical challenge. There is also a cultural challenge here, because would you be happy to read in the label of a device that is going to be used with you the fact that the evidence that was used to produce that device was a simulated or a synthetic evidence? I think you're doing that already every time you jump on a plane, by the way, or you take a car. But there are issues about, what is the quality of evidence that would be required to do this. And what's in conversations that we're having with regulators, the advice is that we should focus first in areas where there are regulatory gaps. There are areas where it's not easy to do trials in the first place, and that's where we should start. And I hope that there will be a not-too-distant future, an opportunity where you have a simulation first opt-out, that you need to demonstrate why you're not doing simulations before you go to do anything on real subjects. And we are doing quite a bit of working with regulators and with the publics and with industry to try to progress this effort through the In-silico UK. So if you're interested in this, you go to insilicouk.org. That's a community that we're building, and Avicenna Alliance is more a European-wide. And I started a few days ago a podcast where we are going to be putting some material from with the spirit of disseminating some of these methods more broadly as well. And with that, I'd like to say thanks to a bunch of people that are working with us and consortiums of companies and collaborators in this space, and also to thank the Royal Academy for their funding and support. Thank you.
Thanks very much, Alex. Questions?
So, obviously amazingly impressive. I just wonder how much would it, or somebody that is coming in with a competing device, would they have access to everything that you've created to be able to test their advice, or would they have to start from scratch? Because, I mean, it's taken you, obviously, many years to build up all that expertise and all the models and everything else. So, yeah, what's that sort of trajectory looking like? So are those models going to be there that people can just access them? Is that the idea? Or
So, okay, so there is a difference between what we have done and what I think we should be doing in the future, okay? So what we have done is like what every other academic has done, which is surviving and trying to make progress with all of this. But if I was able to, what our trajectory is and what we are trying to do now through the In-silico UK network, and there's a group of us around the UK that is trying to develop a center of excellence on in-silico regulatory science and innovation. What we imagine is a public-private partnership that will allow to have those models publicly available, because we believe that in the end of the day, having anatomical populations and having some of these models available will help everyone, right? Because it's like starting at the higher level in your competition, and that will improve the quality of the research as well. Still, there will be a space to compete and to differentiate. The other thing is that perhaps in the future you could imagine some of the, let's say like a training set of those that will be publicly available, but then a set that actually regulators will have separately to test, submissions to the regulatory process, so that you don't have companies just tuning in to the data that is made available. But if you had a data set of 80,000 virtual subjects, I think you start in a completely different position to then go and do a randomized trials. And what I see is the clinical studies will be more confirmatory studies rather than discovery studies, which I think is what they are at the moment, because you will have done a lot of work on these models ahead.
Great. I think you might have been the fastest. You had it up before. Yeah.
Yeah, it's a really fun, interesting, worthwhile problem. A question about, so the simulations you run, the deep learning software, the models, physiological models, they're all written by external modelers, external software, be open source. Is there a risk that it's just kind of kicking the bucket down, the problem down from the hardware engineers to the software engineers, and how do you ensure that?
So that's a good question. So I think I alluded to some of the standards that are being developed. So in particular, if you're interested, the ASME BMB40 standard. So this is the American Society of Mechanical Engineering Validation and Verification standard number 40. These are standards that provide you with the series of steps that you need to follow for demonstrating credibility of a computational model for regulatory purposes. And those are mostly validation and verification ranging from the software all the way to actually compare us against experiments as well. So that's what is required, and that's the rigor that will be required. But that's the rigor that is being used in other sectors in other industrial sectors, like the aerospace, like the automotive. So I think we need to get to that level of quality in the models.
Is there a risk that you still have the same problem where if a model fails and you've blown all this money thinking that your simulation is fine, that you're going to have to spend a lot of money going back to square one?
I think even if I couldn't reduce the cost, but I could improve the quality of the products, I think it would be worth. Because at the moment, the problem we have is that we don't have good quality products, and it's expensive.
Great. I think there was one at the back. Oh, the very back, slightly behind.
Hi, excellent talk. So now that we have various morphologies and also fluid dynamic predictions with various morphologies, are we closer to custom stents for each of the patients?
That's a good one. I think that's a slightly different. So having bespoke devices has other implications. You need to have a manufacturing process that can deliver that bespoke design in a sort of a just-in-time approach, right? Which is different to, now that you have this knowledge, could you come up with a better set of sizes which are standard, but that better cater for the overall population? Those are possible, but they imply have different practical implications. The other thing is, if you have a bespoke system, you might want to use this technique to actually demonstrate that your process of designing performs across the population effectively. Because the minute that you go bespoke, you don't have you can certify every product. You need to certify the process by which you produce them, right? So that also has practical implications on the regulatory side.
Okay, thank you.
I think there is one colleague as well there has been raising the hand.
I'll be quick. Thanks, a really comprehensive talk. It was really great. Something you touched on at the end is how can you increase trust with not only patients but practitioners who are using these devices that are trained on like using these models and using these in silico modeling techniques?
That's a good, I would say that there are two aspects. I mean, the way to do it is like we do in every other aspect of technology, that you need to engage them in the process. This is not something you do to patients, you need to work with them. So we have a patient group that works with us of about 15 people that we met a number of times during the last 6 months in the design of of an application that we submitted a few weeks ago. And you need to do that. The interesting thing is by talking to them, what I found out that what was more difficult for me is to explain why we're not doing it. Yeah. That actually when you start explaining what it implies, an animal experiment and a bench experiment, they have also a lot of questions. So I think the challenge here is how you formulate the questions in a way that you're not biasing the answer, and also that you don't go there with the assumption that what we're doing today is correct. Because they don't fully understand the process. Many of us wouldn't understand many aspects unless we are working specifically in that domain. So I think it's about presenting the information and the facts and then understanding what are the concerns. And most of the concerns that they had was not so much about using modeling and simulation, but about similar questions that they have about AI. What happens with the quality and the representativeness from these virtual populations? Are they going to be biased to a specific subgroups? They would like also the data to be that if data from them is being used, that they're aware about what is being done with it. But these are very similar questions to other AI data usages, I would say.
Should I pass it over?
Wow. Thanks very much for the talk. My question to you is, when you're using animal models or when you're using also clinical trials, one of the ways that you address this scrutiny is by using murine models, for example, that are widely available out there, or also inclusion and exclusion criteria from patients that are widely recognized. In your case, how do you defend when things go wrong, that the problem is the device and not the model itself? Because the model is not widely available and it's the model that's also in development at the same time.
Okay, so I think we need to distinguish what are, let's say, critiques that I would have to all of this approach due to the maturity at this stage, to things that are intrinsic to it, right? I think the wide availability is a bit connected to the first question, and the reason why some of the things are not widely available is because there hasn't been the investment that there has been in other areas to build those populations which are widely available. If they were widely available, then it's similar to animal models. The thing that is, it is recognized that the transferability of a lot of the knowledge in some of these animal experiments is limited. And in fact, there is, when you go, I think it is from phase 2 to phase 3 in most of the trials is where you have the biggest value of death, right? And this is precisely when you go from animals to humans. So I would challenge a bit the assumption that these models are good representations of humans. I think they're good in terms of toxicity and some aspects, perhaps of basic function, but in terms of the continuum, there are disruptions in that. And that's why some people ask, would it be useful to do sort of a virtual mouse model? I say, well, it's a model of a model. Why you bother? You put your energies in modeling a human, right? That is the thing you want to effectively study. So I think it's a bit of a debate in particular. I mean, I'm more familiar with devices than drugs, and the problem with devices is that the anatomy matters, right? So you do a model of a cardiac valve in a mouse, which is, a lot smaller. It's a completely different device. So you do need to really get as soon as possible, as close as possible to the scenario where you're going to be using the devices.